Okay, good morning. So I'm chairing the next 4 sessions before straight to lunch, no coffee. Okay. So I'm going to talk about adenovirus. I'm just going to touch on a few things and we can develop them into a little more detail if at the end. But if there are any burning questions as I go through, I'm more than happy for people to stop me and we can talk about them. We have a little bit of flux in the programme, but we don't want to drift too much because people may come in to talks at a time. So I'm going to talk about adenovirus, some very basic biology of the virus, some of its limitations, its applications in local delivery, systemic delivery. Something I think is really important is use of alternative serotypes. And then lastly, just very quickly, as a tool to probe biological mechanisms, because this may ultimately be the best use of the virus. And I think adenovirus is one of those viruses that really had this huge potential at the start, but there have been a number of problems with it. And we need to learn more about it before it's used most effectively. So in terms of basic biology, Hildegard's already talked about it. It's quite a large virus. It's this DNA virus, and it's been utilised a huge amount in gene therapy applications. The structure of the virus has been fairly well characterised to a high resolution now. And I've put in some of these slides I'm suggesting reading if people are interested in developing or learning more about these particular aspects of the virus. Glenn Nemero and Phoebe Stewart in the US have really made a big impact in this area, understanding the structure and the function of the virus. There are many important proteins involved within the core of the capsid itself as well as on the outer capsid. I'm really going to focus a little bit on the tropism determining parts of the virus. So this is the fiber, the penton base, but also the hexon. So these are the main 3 capsid proteins, and they're all involved in virus infectivity, despite what was originally thought. The other reason why people are interested in the virus is actually making it in the lab. It's pretty easy. They're 36 kilobases or so, plus or minus a little bit. And there are ways of doing this in bacteria, in mammalian systems. There are a bunch of them. It's quite easy. You have to do some large cloning for some of them to generate all the helper plasmids. But generally, unless you've got a really toxic transgene, you can make these pretty effectively. And people use them to probe mechanisms just as much as they do for gene therapy applications. So the most commonly used virus, of course, is adenovirus serotype 5. It's been known for a long time, and there are a number of systems to make the virus, and it's a very flexible system. Just illustrating again the 3 main capsid proteins. So the fiber, this is the fiber shaft or nub domain, which goes into the penton base, which is here in yellow, and the blue is the hexon protein. Now originally the virus, before we had AAVs and all these other much more effective viruses with some applications, and the virus was really being used to cure just about everything you can think of in the early days of gene therapy, even for things like genetic diseases and polygenic diseases such as hypertension, which would never ever do in a clinical setting. It allowed you to probe mechanisms in vivo. And now I think virus particularly attractive in terms of vaccines. I'm not really going to talk about this, but there's a lot of interest in adeno vaccines and a lot of good clinical data coming through. Also for cancer, I'll touch on this, and in cardiovascular disease we just keep going on and on about how useful it can be, but there's been no clinical data yet that's really given us any positive data in the cardiovascular area, which is a little disappointing. We're doing a trial over the next couple of years, hopefully positive, but certainly cardiovascular disease could do with some good news like we have in other diseases. But as yet, I think probing mechanisms like angiogenesis and heart failure and vascular pathology is very difficult using a single intervention. But as Hildegard's touched on, there are many improvements we can make. We can modify the capsid, we can use a completely different serotype, we can modify the genome. I won't talk about that, but you can put this immunogenic virus. You can make helper-dependent viruses by taking out, apart from the ITRs at the end, taking out the rest of the genome so you don't get any expression of the viral genes once it infects itself. So these are helper-dependent viruses, but they're a real pain to make, particularly at the clinical level, and there's very little clinical data on those because making them is such a difficult procedure. As I mentioned, you can use alternative viruses, and although everyone uses Ad5, there are absolutely hundreds of alternative adenoviruses that may be used. I'll come back to that a little later. So just 2 slides on what we do for local gene delivery, and you could use this in any application. You can inject them into tumours, you can inject them into the heart and stuff, and you'll get gene transfer. You have to push it, and it only lasts for maybe a week or 2, unless you use one of these helper-dependent viruses. But essentially, this is what we get. This is an ex vivo application in cardiovascular disease. Patients are undergoing bypass graft surgery, the veins taking out the leg. Surgeons just stick it into some saline for an hour or so before they graft it into the heart. This gives us an opportunity really to do something useful. There are 2 million of these operations worldwide each year, and most of them use this procedure, and 50% of them in the long term fail. So very big clinical need. And we can use an adenovirus 5, we can cannulate the vein as the surgeon throws it into saline in about 2 minutes, and we can infuse it with an adenovirus. The adenovirus is very effective if we use high enough titer. Okay, so this is the only vector system that will work in this setting. Okay. And we get very high levels of gene transfer, and we can do this ex vivo in human, so this is ultimately the target we would use. And we can also do this in pig. This is 7 days after grafting, where it's been explanted, LAC-Z expression throughout the grafted material. This is the artery, with no gene transfer. So this is what the pathology that we're trying to treat. So in this application, through small animal models, through large animal models, we have very nice efficacy using this system, and we're going to test this clinically over the next 2 years. So local gene delivery works very well with an adenovirus, although it's transient, and you have to really force a lot of virions in to make effective levels of gene transfer to do anything useful. And other viruses may be taken over from those applications for liver or cardiac gene delivery. So systemic gene delivery is something I don't think we'll ever do clinically, but we've been interested in it from sort of virology angle. And people are always interested in how the virus, when you inject it into the bloodstream, always ends up in the liver and the spleen. So the liver is a bit of a sink for adenoviruses, and we also get splenic uptake. And there are lots of mechanisms. There's kupffer cell uptake in the liver, there's hepatocyte transduction. And this goes back a long way to some really pivotal work from Glenn Nemero and Tom Wickham. It's published in Cell. That's a long time ago. And that showed, first of all, actually, that the virus gets into cells through integrins. So this is, we go back to the capsid structure, this is the penton base. It has RGD motifs, which bind to integrins, alpha D, beta 3, beta 5 integrins. And that internalizes the virus. At that point, no one knew how the virus actually got onto cells. But then 2 papers a little later, in 1997, showed that this was a Coxsackie adenovirus receptor, which is expressed on the surface, although in tight junctions, which bound to a knob domain, very specific domain on the knob of the fiber. So this is a 2-step process: CAR interaction and internalization via integrins. And this works very well if you have cells in a dish. And of course then we worked out exactly how the virus gets into the liver. But it didn't really work like that. So people started to engineer the virus, and they focused on the fiber protein predominantly, but also the penton. The crystal structure and modeling had shown motifs that were expressed or displayed on the surface of the knob domain of the virus that you could then manipulate with peptides that have been used in the AAV system very well. And for example, this is the HI loop of the virus knob, which really sticks out. And you can obviously imagine that you start to put peptides in here, they're going to be exposed in a way that can interact with the cell. And there are hundreds of papers from Tom Wickham, from our group, from David Curiel, etc., showing this works absolutely beautifully in cell culture. And this is just an example using this very well-known peptide RGD4C, which binds to integrins. It could be a bunch of other peptides popped into the HI loop here. This is compatible with this trimeric fiber assembly without detrimental effect to the processing and folding of the fiber. And this gives you very high increases in the levels of gene transfer, so 10, 20-fold. And also the important thing is it improves the kinetics of uptake of the virus. The virus is actually pretty slow getting into cells, so you can improve both the overall levels of gene transfer as well as the kinetics of uptake. So that's very easy to do, and there are lots of papers on that. The problem came when you started to look at these modifications and look at liver uptake. So the car binding domains in the fibre knob here, the integrin interaction down in the penton. If you mutate these either individually or together, then you had absolutely no effect on uptake of the virus into the liver. So this is a mechanism that's completely independent of these main tropism determining processes that have been published in the late 90s. So work from Dmitry Shegmetov and our group. So Dmitry showed that there's an interaction between the adenovirus fibre and factor 9. That gave us a sniffer of what was really happening. So we found out that the virus is actually binding to this protein, which circulates in our bloodstream. This is factor 10. It's about 10 micrograms per mil. It's very high levels. It has 4 domains. This domain actually interacts with the virus. This is the GLA domain of factor 10, EGF1, EGF2. This is the serine protease domain of factor 10. So this was able to bind to the virus and mediate uptake into cells. And the key thing we showed was actually this was the first activity of the hexon protein in determining tropism. Previously, the hexon protein, which is the most abundant protein on the capsid surface, again a trimeric structure here, was thought to be just a structural protein. But actually, when you decorate the virus with factor 10 and do cryo-electron microscopy, you see factor 10 binds into the cup of the hexon trimer, as you can illustrate here. So this is actually, we've mapped all of this. This is the GLA domain. This is EGF1, EGF2, and the serine protease domain. Sticks out, and actually the serine protease domain, there are lysine motifs in that which bind to peptide sulfate glycans in the liver. So all 3 capsid proteins are now involved in the tropism of adenovirus 5. And one of the key experiments, or 2 of them I'll show you in the next couple of slides. This one we did with a very crude way of modifying the system. So if you inject warfarin into animals, this will block the production of vitamin K-dependent regulation factors. So you dampen down all of those, factor 9, factor 10, protein C, factor 7, and then you can look at whether these proteins are important in liver gene transfer. So we inject these into a mouse, into a rat. We've also done it into microsieves, which is very small non-human primates, and the same mechanism exists. If you pre-treat the animals with warfarin and then inject the virus, you have absolutely no uptake because there's no factor 10 available to then decorate the virus. But if you put factor 10 back in to the warfarin-treated animal just before you inject the virus, you fully restore gene transfer. Okay, so this is really the key mechanism involved in liver gene transfer. And actually, there's some sort of genetic evolution, the phylogenetic tree really bisects viruses, human adenoviruses that do and don't bind factor 10. So most of, or pretty much all of the D viruses have absolutely no binding to factor 10. The ones in blue, the ones we've tested, we haven't tested all of them because they're not all available. And the ones in the other groups seem to all bind to factor 10. And different affinities, but there's definitely something going on in terms of evolution of the virus and its interaction with the coagulation system. So what we did do then was in a more refined way is mutate the hexon. So we focused in on the hypervariable region. So this is modeling of the hexon. So this is embedded in the capsid of the virus. These are the hypervariable loops. There are 7 of them, and they're all in different colors. And these are responsible really for most of the antibodies against viruses targeted against the hexon. And this is very different in all the different viruses, which gives them a different serotype status. So essentially, we mapped all of these with Dan Baruch and Menzo Venga, and we can modify all of these. So this is looking down on the trimer. This is the side view. So if we do that with AD5, which we know binds factor 10, this is a resonance showing you inject the virus over a factor 10 biosensor shift, and you get very good binding. Adenovirus 48, which is a D virus, doesn't bind factor 10 in the hexon. Has actually no binding when you inject over factor 10 shift. And then if you take the capsid of the AD5, put and just swap the regions I just told you about from AD5 into AD48, you eliminate the ability of AD5 to bind to factor 10. So this really mapped that region. And then in vivo we showed, very importantly, that the virus was behaving like this in terms of liver uptake. So So if you take adenovirus 5 and inject directly into the muscle, so that's a process that's driven by the fibre interacting with CAR and or integrins. And then if you inject intravenously at different doses, this is bioluminescence imaging shown through liver uptake at low, medium and high doses. If you take the virus now that we've modified from the previous slide, you completely eliminate liver gene transfer. But you have no effect on muscle uptake. So the virus, when you put it in different situations in the body, you use different capsid proteins to mediate gene transfer. So it all becomes hugely complicated when you inject the virus into the bloodstream. There are interactions with red blood cells. This is mediated through CAR on red blood cells and the knob-domain of the virus. There's interaction with platelets. There's tons of literature on this. Activation of complement system. Most of the virus is taken up and scavenged by Kupffer cells. This doesn't lead to gene transfer. It leads to clearance and death of the Kupffer cells and clearance of the virus very quickly. You get a little bit of endothelial uptake, but the vast majority goes through this factor 10 pathway through heparin sulfate proteoglycans into hepatocytes. But it's a very complicated procedure, and to develop a virus that would only target into hepatocytes and didn't have any of these other effects would be quite challenging. And very recently, Andrew Burns has really shown quite nicely that this isn't the end of the story. So what happens, you talk to Andrew, I think he says it like this, is postdoc injected factor 10 deleted virus into the wrong animals. So these were some immune deficient animals that they had in the lab. And they found that the factor 10 deleted virus actually targeted into the liver, which was a complete shock to them. So they worked backwards to work out what was going on. But essentially, in the immune competent and immune deficient setting, if you have factor 10 circulating, decorating the virus as you'd expect, then you get gene transfer quite effectively into the liver of both types of animals. But if you have a factor 10 deleted virus, and you can do that with warfarin, or you can genetically modify the virus, in the immune competent setting, you get active IgM targets into exactly the same regions where factor 10 binds. So if you have no factor 10, IgM will bind in. It's sort of a selective but non-selective way and activate the complement system, which neutralizes the virus. And this is eliminated if you have animals that have an insufficient immune system. So this then starts to get very complicated about adenovirus and immune interactions. And I'll talk a little bit about more about that in some recent work tomorrow. But it's complicated. So I just want to touch on some alternate serotypes of viruses because these are very important. There is a lot of work on using alternative human serotypes, particularly in the cancer setting. So using CD46 as a receptor. A lot of the B viruses, Ad35 for example, use CD46 as a receptor. There's desmoglein-2. There's also other receptors that the virus uses, but they categorize them. So then you can target the virus at different cell types. CD46 is a bit of a pain because it's not expressed essentially in neurine systems. But there's a lot of interest in using that as targeting alternative receptors in different cell types, as well as other viruses. And there are some non-human serotypes that people are starting to really think about. And there's a lot of work in using chimpanzee adenoviruses in vaccination, using alternative human viruses. Dan Baruch's got a lot of clinical trials in conjunction with Purcell, but also on his own, one using Ad26 for HIV vaccination as well as other procedures. So there's a lot of interest also in non-human serotypes of the virus. So touching on that pretty briefly, so that if you just very simply look at the human adenovirus family, there are 60 plus serotypes of them now in 7 different subgroups. The problems with Ad5 I've already stated for a sort of gene therapy setting. There's a lot of pre-existing immunity. It varies across the world. Some places it's 80%, some places it's 30%, but it's very high. And this is actually one of the problems with some of the AAVs as well. So the recent Cupid trial in cardiovascular for heart failure really had to, using an AAV1 virus, had to exclude up to 70% of patients who had pre-existing immunity against the AAV1. So this is a problem for a lot of viruses. So we can avoid that by using alternative viruses that maybe are much rarer globally and low infectivity rates. And of course, there was a clinical trial death in 1999 that everybody knows about, that really highlighted that we need to understand a lot more about viruses and maybe think about using alternative ones. We've been interested in cardiovascular disease because Ad5's okay, it's not brilliant, but we can use alternative serotypes for vascular gene transfer. So this is an important area, but the problem is that it's very difficult to know which virus to use. How would you select it? And actually, it's very difficult to make vectors from these things. So it's very easy to get serotype from ATCC or wherever, or just get it from one of the guys that isolates them. But you can't really use a replicating virus like that to assess gene transfer. You need a vector system that expresses GFP or XZ to make it nice and easy for you. And there are very few available from these, and this is one of the problems that we need to try and deal with. Just one example, we've been very interested in adenovirus 49. There's 3 papers on this since it was isolated, and it's a very rare adenovirus. And we were interested in it because when we screened adenoviruses for uptake into cardiovascular endothelial cells, it was very high. So we don't know how it does it. We're working on the receptor at the moment, but essentially it could be very useful. And actually, when you look at pre-existing immunity in a Glasgow population, so we just got plundered on serum from cardiovascular clinics, the high neutralizers against Ad5 is about 30% in Glasgow. It's pretty low. But if you look at Ad49, it's absolutely no. So nobody has ever seen this virus. We've not found serum yet that neutralizes Ad49. So combining its tropism for vascular cells as well as its profile with very little or no pre-existing immunity makes it very attractive. And we'll work on that virus in the future. Lance Seymour gave me a couple of slides just to illustrate how they're modifying adenovirus and using alternative serotypes. They're very interested in virotherapy. Conceptually, this is nice, of course, because you have developed, genetically engineer, isolate, whatever, an adenovirus that selectively is taken and spread through tumour cells and selectively killed them and keeps normal cells magically okay. And you can do this by combinations of uptake mechanisms, transport mechanisms, spread mechanisms, as well as mechanisms to control virus replication in normal cells versus tumour cells. So this has been really a goal for many years, so you can destroy the tumour by the virus and leave everything else lovely. And the way they've done this is, and they've coined this anadotuxirav, easy to say, but essentially this is a virus they're working on. And they were very clever about the way they did this. So they took cancer lines in vitro, and they basically threw a lot of viruses in. So these were viruses that were available and selected for their potential to be taken up into cancer cells. And there's quite a lot of pre-existing data to show that some of these alternate viruses have capsid host interactions that would be favourable. You can throw them in. These are replicating viruses. You stick them in at very high multiplicities of infection. It really selects a profile that you can begin to invite recombinations between these different serotypes between these genomes, and then kind of evolve by force of evolution a virus that comes through these different panning procedures and to give you viruses that have a genomic structure that allows very rapid replication and cell death, lysis in these tumour cells. So that's exactly what he did. And you can ask Len because he's here, but essentially they're doing all the clinical trials with this virus now. It works very well in cell culture, it works very well in animal models. And from their first sort of clinical data, where they've been had access to tumours, they get very high levels of viral load in these tumours. So it looks as if also this works in the context of human cancer. And one of the problems, of course, we're working between preclinical and clinical models is a lot of the adenovirus receptors are slightly different in terms of distribution. The replication is different. In mouse, you don't replicate adenoviruses very effectively unless you use a mouse adenovirus. So transiting through preclinical models into the clinical setting is often quite difficult. So this is actually very exciting, and this could also be used as well as a therapy, but also used as a diagnostic, because you can then modify the genome and structure of the virus to allow you to do all these sorts of things if you can selectively target into the tumour. But alas, that's all human adenoviruses. So we're working with some Hungarians and Swedes on non-human and other human adenoviruses, and there's an enormous amount of scope out there, both in terms of human-related viruses and mammalian viruses, as well as in other species. And you may think that's a bit wacky, but there's some really interesting stuff. We're actually working on a snake virus that actually has 3 proteins. So while that just sounds a bit odd, we're interested in why anything would evolve with 3 different fiber proteins and how they interact at the cell surface is really quite novel. So that's the only virus that we know has 3 fibers. So from a sort of geeky science point of view, it's quite interesting. Whether we use it clinically, we don't know, but we're just currently making a vector system from that. So there are about 162 very accurate, fully sequenced types, and these are across all of these species that you can think about using in terms of not necessarily in gene therapy, but using in terms of to sort of dissect virus receptor, virus host interactions. And as I said earlier, the sort of the primate adenoviruses, and there are many of these as well. These are all sort of genome sequenced. There's a lot of intellectual property around these, and there's a lot of interest, particularly in vaccine development. So adenovaccines, as I mentioned earlier, are pretty topical. One of the things that we're doing is I'm coordinating a couple of training networks. Well, this is the training network across Europe using adenoviruses in vaccine development, cancer, there's also cardiovascular. But this one here is an industry academic partnering program where we're taking some of these weird and wonderful non-human adenoviruses, making vectors out of them, and seeing what we can do with them in terms of gene therapy and vaccine development. So finally, just using adenovirus as a tool, so some really neat work on this non-human adenovirus that is neurotropic. It's called canine adenovirus 2, so it's a dog virus. And Eric Kramer in Montpellier has really pioneered this field. So he's interested in canine adenovirus 2. This is looking down on the fiber of the penton structure. This virus is a bit weird. It has a fiber that's pretty long. It has 2 hinge regions on it, and it's thought that this gives it really exquisite flexibility once it interacts with this receptor. So it's a bit weird because the Ad5 doesn't, it interacts with CAR, but it's really just as an engagement receptor. It can internalize directly through CAR, that's not really being published. But this canine adenovirus 2 combined to CAR and be completely, exquisitely internalized with it. And this then traffics through neurons and allows gene transfer into other parts of the brain. I'll show you that in a second. And it's all through its ability to interact with CAR. It also binds sialic acid, and the crystal structure of the interaction with CAR has all been developed. And there's a helper-dependent version of this virus as well. So what Eric and colleagues have shown is that if you inject into the striatum, you get this CAR-mediated trafficking, and you can find the virus and gene transfer in all these other areas of the brain. So this allows you actually to use the virus as a tool to map neural networks. And this is obviously very topical at the moment. You can do this in rodent, he's done it in dog, and he's doing it in primates as well. So he's developing this as a virus for gene therapy. Whether it be better than AAV, who knows, but canine adenovirus gives you very good volume. So this is just mapping the volume of gene transfer from a single intracranial injection of the virus. This is using a helper-dependent virus. It's probably actually much more difficult to make clinically than AAV would be, but nevertheless it gives you beautiful brain gene transfer. And he's used this as therapy with colleagues targeting lysosomal storage disease, NPN7 in dog model. This is a helper-dependent virus. You've got to know a lot about making canine adenoviruses to be able to make a helper-dependent canine adenovirus. No idea how he's done it, but you have to use a GMP dog line, which is infected with the virus. This has been really very useful. And he's shown then with colleagues in MPS7 dogs that if you give this intracranial injection of this helper-dependent virus, you get really beautiful gene transfer in the brain, and this really corrects the neuropathology in MPS7 dogs. So this is MPS, and you get these backdoor-like structures here, as you can see. This is corrected by the helper-dependent virus and really looks like a healthy brain. And this is in the choroid plexus, as you can see. This is healthy. This is the MPS 7, and this is the restored. So it works very effectively, and you can just read these papers if you want to understand it a little bit more. But also, what this has done, and there's a recent Nature and also a Cell paper on this in the last couple of months. They've used the canine adenovirus as a tool here to inject into the brain, but also to map neural networks in the brain and actually understand a lot about these processes. So you can actually take these brains and you can identify cells that have been infected through these neural networks, and then detail some fundamental biology. So these sort of unpredicted infectivities that you see from these viruses allow you to do new stuff, not necessarily as a gene therapy vector, but also to probe these biological mechanisms. And I'm going to stop there. I'll take any questions. Just thank Len Seymour, Eric Kremer, and Balash Harat for providing me with some of those slides I've used today. So, thank you.
Quick question, curiosity. The clinical trial or this vivo experiment is it done with hyper-independent or first-generation vectors, and how are you planning them to do the clinical?
So all of our data preclinically is in the human ex vivo model and the pig in vivo model, and we've done it all with first-generation viruses. So we're overexpressing. The theory there is that when you put a venous graft into the arterial circulation, it's a very early remodeling that predicts or underpins the late failure of those grafts. So the theory is that just a short time of inhibition will give you a downstream signal that's positive. So we in the pig model, we've looked long term, the virus has completely disappeared, it's immunogenic, but we get lasting effects on remodeling. So at least in the pig, then it's effective. Whether it works in human, the kinetics of neoinsulin formation and vascular modeling may be a little different. We have tried to make a helper-dependent TIM3 virus because that's our transgene, but it's hell on earth. So we're going to try with the first generation virus, which is also GMP production, is hell on earth as well. So we'll get there. Any other